First, we will present about how to make a herbarium. Step 1, collecting.
And Assalamualaikum, my name is Norerliana Syafika. Okay, the first step is to look for plant specimen that have a perfect structure that has flower, leaf, root, stem and root. We have collected plant specimen from the described location. We carefully select healthy and perfect plant. So enjoy the video.
After VBC, there is two plants that we collect for our herbarium: the Melastoma melibacterium and Siluria ternate. Step 2, pressing.
So now we will move to the next step. To create a herbarium, the collected flower, which is Singaporean seludok, flower are arranged on a press board and covered with newspaper. Adjust the position of flower's head so that it doesn't come off the presser board. The position of the leaf also need to be alternated between the front and the back of the leaf to allow the identification process to be done more easily. Make sure the flower taken are complete with flower, leaf, stem and roots so that the herbarium is complete. Once the flowers are in the right position, cover them again with newspaper and pressing board. All the steps above are repeated for the bunga telang. Both flowers are overlapped and placed in the pressing board. Tighten the pressing board with a rope. Place the pressing board under a heavy object and leave it for 2 weeks.
Step 3, mounting. For the next step is we have to sewing the herbarium to look that herbarium look nice and properly. First at all, we take out our herbarium from the pressing board. So we start the sewing at the needed part of the plant. For our herbarium plant, we sewing at the part of root, stem and the top of the plant.
After the flower process is dried, apply a little glue on part of the leaf that you want to paste so that the leaf do not fall.
After that, for the next mounting is take a glue paper and put the water and put at the back of herbarium to close all the bad thread on the herbarium. Step 4, labeling. For the next step, we must to take an identification paper and write all the information of that herbarium. The information needed is scientific name, local name, locality, coordinate, and others.
The last part is to paste the identified paper on the right side using glue.
So that's all of the steps of make a herbarium plant. Next, we go to the step flower dissection.
Here are the steps to perform flower dissection. First, we must remove the sepals and the petals. Gently hold the base of flowers and remove the outermost part, which is usually made out of sepals. These are the small green leaf-like structures at the base of the flower. Next, carefully remove the petals by cutting them off where they are attached to the base of the flowers. The next step is we must remove the stamen, which is the stamen is the main reproductive structures of the flower. It consists of a long, slender filament with an anther at the top. Carefully cut away the petals to expose the stamen. The use of forceps is necessary to handle the delicate parts. Examine the anther and look for powdery pollen grain inside.
This is our labeling after the dissection.
The next step is dissection fruit from flower tree Singapore rhododendron. As you can see, we split the fruit using the scalpel. To do this method, we need to be calm and slow because we do not want to damage the fruit. First, we split the fruit in the middle to show the condition inside the fruit. Here, we can see the exocarp, mesocarp, endocarp, and seed parts of the fruit. The exocarp is the outer layer of the fruit. The mesocarp is the inner part of the fruit. The endocarp is the outer layer of the seed, and the seed for this fruit is the small white seed part. The green stem is a pedicel, which is a fruit stalk. The second fruit is the fruit of the telang flower tree. In contrast to the condition of the fruit from the Singapore gourd flower, the fruit from the telang flower tree is long and green in color. This fruit is split in the middle. After splitting similar to the state of the fruit before, we can see the exocarp and endocarp of this fruit. But the difference is this seed can be seen clearly and counted. This seed has 8 seeds.
So here is the result of dissection of the fruit. That's all from our group.